Welcome to this lecture on the biochemistry of metabolic processes. Today, we are going to explore the liver, an organ that acts as the ultimate metabolic command center of your body. Imagine a bustling city with a massive central train station that controls all the cargo moving in and out. In your body, the liver is that central train station, and the cargo consists of the nutrients you eat. Let us dive in and see how it manages this incredible task. Now that we have our train station analogy, let us look at how the liver actually handles incoming cargo. When you consume a meal, all the sugars, fats, and proteins do not just float aimlessly. They travel straight to the liver first through the portal vein. The liver acts as a brilliant traffic controller. It constantly makes critical decisions. Should it store these nutrients for later, burn them for immediate energy, or send them out to other organs, like the brain and muscles? It is the ultimate chemical factory, constantly adapting its machinery based on whether you just ate a huge feast or have been fasting for days. Building on its role as a traffic controller, the liver's primary job, in biochemical terms, is maintaining systemic metabolic homeostasis. What does this mean? It means keeping the blood concentrations of circulating fuels, primarily glucose, fatty acids, and ketone bodies, perfectly balanced. The liver cells, which we call hepatocytes, are uniquely equipped for this. They contain a comprehensive set of enzymes that allow them to perform a wide array of processes: glycolysis, gluconeogenesis, glycogenesis, glycogenolysis, lipid synthesis, beta oxidation, and the urea cycle. This incredible versatility is what keeps our body's energy levels stable throughout the day. To manage all those complex pathways we just discussed, the liver relies on a remarkably elegant system at the molecular level. At the core of this metabolic flexibility are three critical junction molecules: glucose 6-phosphate, pyruvate, and acetyl-CoA. Think of these as the main switching yards in our train station. Depending on the hormonal signals received from insulin and glucagon, the liver routes these central metabolites into different pathways. If insulin is high, they are used to build complex molecules for storage. If glucagon is high, they are broken down to release energy. It is a beautifully coordinated system. Understanding these junction molecules helps us appreciate why the liver's metabolic role cannot be overstated. Quite simply, without it, human survival between meals would be impossible. Consider your brain. It relies almost exclusively on a steady supply of glucose to function. When you are not eating, it is the liver's job to synthesize and release this glucose into the blood. Furthermore, our bodies constantly break down proteins, which releases toxic ammonia. The liver protects us from fatal neurological damage by safely converting this ammonia into urea for excretion. It is constantly working to keep us alive. While the liver handles short fasts between meals easily, what happens during prolonged starvation? When glucose reserves run out, the liver steps up to save the body by converting fatty acids into ketone bodies. These ketone bodies serve as a crucial alternative emergency fuel for the brain. By providing this alternative energy source, the liver prevents the body from rapidly breaking down essential muscle tissue to make glucose. This adaptation is a perfect example of the liver's ability to shift its metabolic machinery to ensure our survival under extreme conditions. We have seen the amazing things the liver can do, but this brings us to an important question. How does the liver actually know which metabolic pathways to turn on or off at any given moment? To understand this, we must look at its internal energy status. The liver acts as a highly sensitive sensor, constantly measuring the ratio of high-energy molecules to low-energy molecules within its own cells. This internal energy charge tells the liver whether it has enough surplus energy to build things, like synthesizing glycogen or fats, or if it needs to break things down to generate more energy for itself. This concept of sensing energy is mathematically represented by the adenylate energy charge equation. Let us break down where this formula comes from. The cell's energy is carried in phosphate bonds. ATP is the primary energy currency, carrying 2 high-energy bonds. ADP has only 1, so it counts for half as much, which is why we multiply it by 0.5 in the numerator. AMP has none. The denominator represents the total adenylate pool. So this formula calculates the fraction of the total pool that carries high energy bonds. The value ranges from 0, meaning all AMP and no energy, to 1, meaning all ATP and full energy. When the energy charge approaches 1, the liver turns on energy-consuming anabolic pathways. When it drops, it turns on energy-producing catabolic pathways. Now that we have our formula, let us see this with a concrete example. Imagine a patient has just consumed a large carbohydrate-rich meal. We measure the adenylate concentrations inside a liver hepatocyte and find ATP is 4.0 millimolar, ADP is 0.8 millimolar, and AMP is 0.2 millimolar. We need to calculate the energy charge to determine the metabolic state. We substitute these given values directly into our formula. In the numerator, we take the full value of ATP and add half the value of ADP, giving us 4.4. In the denominator, we sum all 3 to find the total pool size, which is 5.0. Dividing these gives a final energy charge of 0.88. Because this value is very close to 1, it indicates a high energy state. The liver will use this abundant energy to store excess glucose as glycogen. We just saw how the liver behaves after a meal, when energy is high. But what happens during fasting? As we discussed earlier, the liver must synthesize glucose for the brain through a process called gluconeogenesis. However, building glucose from scratch requires a massive amount of ATP. Where does the liver get this energy when you are not eating? It gets it by burning fats. Specifically, the liver performs beta oxidation on fatty acids like palmitate, a 16-carbon saturated fatty acid. By breaking down these fats, the liver generates the abundant ATP needed to drive gluconeogenesis and keep the brain supplied with glucose. To understand exactly how much energy a fatty acid provides, we use the net ATP yield equation. Conceptually, this formula simply adds up all the energy generated by the breakdown products and subtracts the initial investment required to start the process. The first term represents the ATP generated when acetyl-CoA enters the citric acid cycle. The second term represents the ATP produced by the electron carriers, NADH and FADH2, when they enter the electron transport chain. Finally, before a fatty acid can be oxidized, it must be activated by attaching it to coenzyme A. This activation consumes energy, so we must subtract this cost to find the true net yield. If the number of carbon atoms in the fatty acid increases, the yield of acetyl-CoA and electron carriers will also increase, resulting in a higher net ATP. Let us apply this formula to calculate the total net ATP yield from 1 molecule of palmitate. We are given that breaking down this 16-carbon chain produces 8 acetyl-CoA, 7 NADH, and 7 FADH2, with an activation cost of 2 ATP. First, we calculate the energy from acetyl-CoA. Each yields 10 ATP in the citric acid cycle, so 8 times 10 gives us 80 ATP. Next, we calculate the energy from the electron carriers. 7 NADH, yielding 2.5 each, gives 17.5, and 7 FADH2, yielding 1.5 each, gives 10.5. Adding these gives 28 ATP. We sum the 80 and 28 to get a gross total of 108 ATP. Finally, we subtract the activation cost of 2 ATP. Our final net yield is a massive 106 ATP. This perfectly illustrates why the liver relies on fatty acid oxidation to drive the energy-expensive process of gluconeogenesis. Now that we have calculated the energy yields, let us visualize how all these processes connect in this schematic of a liver hepatocyte. Look at the center. You can see our 3 critical junction molecules: glucose 6-phosphate, pyruvate, and acetyl-CoA. Notice how glucose enters the cell and is converted to glucose 6-phosphate. From there, it can be stored as glycogen at the top or broken down through glycolysis to pyruvate and then acetyl-CoA. On the left, fatty acids enter and undergo beta oxidation to also become acetyl-CoA. This acetyl-CoA can either enter the citric acid cycle to produce the ATP we just calculated, or it can be converted into ketone bodies. Finally, observe the export arrows. The liver sends glucose and ketone bodies out to power the brain and muscles. This diagram beautifully captures the liver's role as a central metabolic hub. We have covered a lot of ground today, so let us reinforce the most important ideas. First, the liver is the undisputed master regulator of human metabolism, processing carbohydrates, lipids, and proteins. Second, it acts as a highly intelligent sensor, monitoring its own adenylate energy charge to seamlessly shift between storing nutrients after a meal and mobilizing them during a fast. Finally, by interconnecting key metabolites like glucose 6-phosphate, pyruvate, and acetyl-CoA, the liver ensures that all organs, especially the brain, have a constant and reliable supply of energy, regardless of our eating habits. You now have a solid understanding of how the liver functions as the ultimate metabolic hub. Great job following along with these complex pathways.